All right, everyone, we're going to go ahead and get started now. So again, first and foremost, thank you so much for attending today's Spotlight session. My name is Sim Sliney. I'm a Principal Systems Engineer for the Digital LightCycler system with a background in oncology and NIPT assay development. And I'm very excited to discuss the Roche Digital LightCycler system with you today. And so for today's session, I'm going to go ahead and cover and begin with what is digital PCR, just to level set, for the audience, then followed by a brief overview of the Roche Digital LightCycler system. It's a powerful new addition to the Roche PCR ecosystem. And lastly, present some data on an external evaluation of the Roche Digital LightCycler system performed by several sites around the world. And so let's begin with what is digital PCR. So the fundamentals principle of dPCR actually lies within the capability of partitioning, essentially, which permits absolute quantification. And how this is accomplished, essentially, is first by preparing your reaction mix right here on the very far left-hand side. And this is inclusive of your master mix, your sample, your primers, and your probes. And then actually partitioning this sample into numerous small volume chambers in which molecules are essentially randomly distributed in each of the compartments. And you can have zero or the absence of a template in a compartment. You can have one or many molecules as well. After the partitioning occurs, there's a thermal cycling and an endpoint detection which occurs, and this essentially is plotted where you have on the x-axis your partition number, on the y-axis your intensity levels, and you can imagine that partitions that had no amplification will be at a lower fluorescence intensity relative to partitions that had template and therefore generated a positive signal, which will have a higher fluorescence intensity. And essentially, there is a classification of your negatives and your positives by drawing a threshold, which allows for you then to apply Poisson calculation to determine the actual concentration. And that's necessary in order to compensate for if you had more than one molecule per partition. So accordingly, the key advantages of digital PCR actually kind of lie in the fact that you have this absolute quantitation capability, again, without a reference standard, which is usually the case, for instance, in conventional qPCR. Now, the other attribute and a key advantage of digital PCR is they're able to detect at an increased sensitivity, especially for low abundance targets. And this is just kind of depicted here on the left here as an example. Let's just say hypothetically you have a 1:8 mutant to wild type ratio in a 16 microliter sample. When you kind of take this bulk sample and actually partition it into smaller volume reactions, there is an enrichment of your mutant and a capability, therefore, to detect it outside of the wild type background. Again, really much increasing the sensitivity. In addition to that, the other attributes or advantages of digital PCR include the fact that you have a really nice high precision capability over a large dynamic range with more partitions, 100,000 versus 20,000, giving even higher precision. And this is depicted here on the right, where on the x-axis, you actually have what we call lambda, or the number of copies per partition. And on the y-axis is essentially your coefficient of variation. You want this to be as low as possible to ensure that you have a very precise measurement. In blue, we're representing 100,000 partitions, and in green, we're looking at 20,000 partitions. And I think what you can appreciate is that over a larger dynamic range, so inputs going from 0 or 1 lambda all the way up to a lambda of 6, that you have a lower CV with more partitions present over a larger range of inputs. Now, these key advantages actually make digital PCR a vital tool for clinical research, including oncology. And while there are existing dPCR systems on market, we at Roche really wanted to design a dPCR system that would allow users to really push the boundaries of their research forward by filling in the gaps and making improvements upon existing offerings. And accordingly, we've incorporated a unique combination of technical advancements. And these include, or are best remembered by the days of the year. So in this case, we're looking at 365. 3 different nanowell configurations that can be tailored to your specific application of need. 6 advanced optical channels. There's actually another or 7th channel which we use for control, but effectively 6 channels that content can be placed into, with the additional channel actually being used for a filling dye that allows for even more precision in measurement. And then 5 being the concentration of the actual DNA and or RNA master mix, which enables increased sample put due to the fact that it's highly concentrated. Now, these technical advancements have been collectively integrated into what we call the Roche Digital LightCycler system. And that is kind of depicted here, which includes the analyzer, as I mentioned already, which has 6 optical channels that allows for multiplexing capabilities, a standalone partitioning engine, which can be placed in a pre-PCR lab as opposed to kind of having a separation to minimize contamination from a post-PCR lab, essentially that allows for uniform distribution of the sample as it's being partitioned into the consumable itself. There are the generic DNA and RNA master mixes, as I already mentioned, have been highly concentrated to 5x and also have been maximized for multiplexing capabilities. And finally, the 8-well, 9-well plate, essentially, which is kind of located here. And you can see that there are 3 different configurations. Again, can come with varying partition sizes and fill volumes. Here we have the high resolution with a 15 microliter fill volume and 100,000 partitions. The universal, which has 30 microliter input volume and about 28,000 partitions. And finally, the high sensitivity with a 45 microliter input volume and about 20,000 partitions. Now, each of these nanowell plates actually can be tailored for your specific application of interest. And what I mean by that is, for instance, you can imagine that having more partitions is really necessary for having precise measurements, which is the case for things like copy number variation, where you're looking for small differences in copies, distinguishing copies. The other attribute of this is another kind of example is the high sensitivity, where you have a large input volume and about 20,000 partitions. So you can imagine that this might be very important for things like rare mutant detection or measuring MRD, for instance, where there's a need for high sensitivity, given that there are minimal copies already present in the sample itself. Now, the digital LightCycler system itself has an enhanced ability to detect these minimal copies of DNA and RNA molecules due to, for instance, as I mentioned, the high sensitivity consumable, but also coupled with the fact that we have a very highly concentrated master mix at 5x. So you can ensure that you have a lot more sample that's being actually measured. Here's just kind of showing that sample efficiency in practice and compared to a very familiar on-market competitor. You can see that with the usage of the high sensitivity plate, with a 45 microliter input volume, you can imagine that with regards to utilization of a 5x mix, there's an opportunity to essentially measure about 31.5 microliters of your sample, right, which is a lot, and which is really important when you're looking for minimal amounts of nucleic acid. In contrast to the 20 microliter reaction with a 2x kind of concentrated master mix, where only 8 microliters of the actual sample can be measured. This is almost 4 times as much sample in a single lane that can be measured on this system. Now, additionally, when that's not depicted here specifically, is that the fact that we have 6 optical channels, which allows you to assess multiple targets within a single sample reaction, right? And this is, again, collectively, these features ensure that when you're looking at sample on the Roche Digital LightCycler system, that you have optimal sample usage. So now that I kind of have you a little bit more familiarized with the system components, let's move on to the actual workflow. And there are 2 types of workflows that can occur on the Digital LightCycler system. One is more research-based, and the other is a little bit more diagnostic-based. And I'm going to focus on the diagnostic in particular. So in the diagnostic workflow, first we have samples that are run and entered into a web application, either manually or through a lab's kind of LIS system. And then it's associated with a particularly bar-coded nanowell plate. And that means that at the outset, the sample actually is fully positively tracked throughout the entirety of the workflow. So once that assignment occurs, you can imagine that you have your extracted sample. We are compatible with FFPET, cell-free nucleic acid, whole blood, stool, as well as swab. You can take that extracted sample, go ahead and prepare it with your reaction mix, which essentially includes your 5x digital PCR master mix along with your primers and probes. And then essentially take that sample, manually pipette it into the consumable, which has 8 lanes. So you can imagine a sample per lane effectively. And then that plate is then taken and placed into a partitioning engine. Again, this partitioning engine is a standalone in the event that you'd like to ensure that you have separation between a pre and a post-amplification lab. You can imagine that the partitioning can occur in a pre-amplification lab to avoid any type of amplicon contamination. And this particular partitioning engine will essentially allow, by a pressurized based mechanism, to input oil as well as allow for the sample to kind of go across the entirety of the lane in such a manner that everything is sealed. So it's a very nice, controlled, contained system. And this is done one at a time, so one plate is partitioned at a time and can take anywhere from 2 to 4 minutes, depending upon the plate type that's utilized. Once the plates have been partitioned, they can then be placed on the analyzer, and once they're loaded on the analyzer, this is where we have both thermal cycling and that endpoint detection, along with the imaging occurring in a bifurcated manner. The analyzer can accommodate 12 plates, so you can imagine that's anywhere from 8 samples on a given plate, so 1 plate to 12 plates would make a batch size of anywhere from 8 to 96 samples in a single run. And you can also have mixed plates within a given run as well. So you can put a combination of high sensitivity plates along with high res plates, as long as they're utilizing the same thermal cycling profile. Once the analysis or the analyze has kind of completed here, the data results and the data outputs can be kind of shared with either the LIS, the web application, or actually opened up in the development software for further downstream analysis. And now just to kind of walk you through, since we will be seeing a lot of these plots in subsequent slides, in the development software, the data outputs include the 1D scatter plot, which is kind of shown here on the left, as well as 2D scatter plots, 2 different types, one kind of reflective of a non-competing duplex, the other kind of reflective of a competing duplex. And just to kind of walk you through this, for a 1D scatter plot, essentially what you're looking at is fluorescence intensities on the y-axis, your partition kind of numbers here on the x-axis, and those that were classified as negatives or had no target present are classified as negatives, and those that had target present were classified as positives. And again, this is just thresholded, either within the development software automatically or can also be done manually. So there's definitely options in the development software to do different types of thresholding capabilities. Now with the 2D scatter plot, this is looking at 2 different channels. So for instance, in cases where you have 2 targets present in 2 separate channels, so FAM and HEX, there's an opportunity to look at all clusters, together in a 2D space. And it's often used when you have mutation and or, kind of indel assays to help with the thresholding capabilities. Again, with the non-competing, you can see 4 separate clusters, 1 negative cluster associated with 1 channel and the positive, again, with 1 channel on one axis, and on the other axis, you have your negatives as well as your positives in the other channel, and effectively your double positive population. Now, once these thresholds have occurred in the development software, essentially this allows for quantification, right? The absolute quantification, because you have these classifications and counting of your positives and negatives after Poisson is kind of established. And this is kind of a representation of the output data tables that come out of the development software that actually gives you your quantification methods. So again, up until now, I've kind of shown you the features of the system itself, the workflow, and the data outputs. And so now I'd like to kind of transition and really talk about this external evaluation, because at the end of the day, what really matters is how the system performs. And so to this end, we really wanted to test how the performance of the Digital LightCycler system, is. And so we did this by conducting an evaluation across 7 sites throughout the world. And the evaluation was essentially designed using 2 representative oncology assays. And this included the rare mutation assay, BRAF V600E, as well as a copy number variation assay, which is a HER2 with the balancer of EIF5 in the FAM channel. Now, in this case, and just to kind of highlight this, and I will kind of mention this throughout the presentation, for the BRAF V600E, which again is representative of the rare mutant assay, it was actually an off-the-shelf Bio-Rad assay that was utilized, on the Roche Digital LightCycler system. So there was no optimization for this assay specifically for the Roche Digital LightCycler system. It was commercially available from Bio-Rad. By contrast, for the CNV assay, we actually used an NIST published oligomer design. And NIST, for those of you that are not familiar, is just the National Institutes of Standards and Technology. So they had a publication for this particular design that was then utilized for this particular study. Now, if we focus on the first experiment, I know it's a little bit of a busy slide, so I kind of want to just showcase this right here specifically. You can see that the primary goal essentially for experiment 1 was essentially to understand how accurate and reproducible the quantification was across multiple runs within a site, as well as across all sites that were included in this actual evaluation study. In addition to that, when evaluators actually had another platform already housed within their lab, there was also an opportunity to look at that across those other platforms, and in certain cases we'll be able to showcase that data as well from a qualitative standpoint. So let's go ahead and look at what's depicted here as the results. So for the first experiment, as I mentioned, we're looking at the rare mutation assay, again, BRAF V600E, an off-the-shelf Bio-Rad commercially available assay. And what we noticed here is that across all the 7 sites, which was depicted here on the x-axis, which are labeled here, you can see that we have sites kind of ranging from the US, from China, Germany, all the way from Japan as well. And on the top panel, what we have depicted here is essentially the sample concentration in copies per microliter. And below it is the mutant allele fraction as represented in a percent. Now, on the very far left of this panel, you can see that we're looking at the 100% wild type sample. And on the far right is the 1% mutant sample in comparison. Now, I know it's a little bit difficult to see, but the other thing to kind of note is that based on the manufacturer's kind of stipulated values, we have that dotted here in lines here, and that's kind of highlighted here with the text. Expected wild type is listed here. The expected mutant is listed here and kind of bifurcated here with this line. And again, the expected mutant allele fraction is also depicted by this gray line. And all sites are color-coded. And what I hope you can appreciate is that, for the wild-type sample, as well as for the mutant sample, across all 7 sites, across all runs performed by each site, we have highly reproducible results that are consistent with what is expected based on the manufacturer's kind of specified values, right? Again, here we're looking at, multiple runs and getting the expected results, which is really, really great. I do want to point out that in the wild type, we did have some false positives that were present, but you can see that the false positives were very, very small and very minor. At the worst case scenario, we had about 1.52 copies per microliter. So what the take home and the key finding from this study essentially is that the Roche Digital LightCycler system really demonstrates the consistency and reproducibility, across different testing sites. And I just want to kind of extrapolate from that and indicate that, these are different users, these are different systems placed across, the globe essentially, but they all are generating on the Digital LightCycler system the same result, which is really, really powerful. Essentially that means that if you were to take a Digital LightCycler system, you can expect very, very high quality results in terms of quantification, as well as very, very reproducible results, which is again demonstrated by this study. Now moving on to experiment number 2. In this experiment, the goal essentially was to understand, what is the potential analytical sensitivity of the Digital LightCycler, right? Obviously, we had a fixed input from experiment 1. We want to kind of showcase what is it like as you go close to your LOD, for instance, right? So we just want to get an idea of what that analytical sensitivity looks like. And in this case, what we're using again still is the Bio-Rad off-the-shelf BRAF V600E assay, essentially, again, representative of a rare mutation assay. And again, very similar to the plots that were previously shown for experiment 1. On the very top panel, we're looking at concentration, which is copies per microliter. And on the bottom panel, we're looking at mutant allele fraction represented as a percent. Here we have the sample percentage in terms of the mutant contribution. You can see that we're going all the way from about 0.02% all the way about to 85% here. And wild type obviously having no mutant present, so 0% mutant there. Now, what I'd like you to appreciate is, again, all 7 sites are present here, color-coded, and you can see that across the 7 sites, we have really great analytical sensitivity at all sites. Again, the estimated analytical sensitivity was less than 0.1% with an R-squared above 0.99 in the serial titration that was performed, essentially, with the false positive rate being very, very low. Again, that's indicative here of the wild type, with only about, worst case scenario, 1.54 copies per microliter. So essentially, what we can take home from this is the fact that, because of the false positive that we do see present here, that when we look at the capability of what the analytical sensitivity is, it's actually going to be around 0.07%, right? Because we want to be at least 2 standard deviations away from where we do see the wild type kind of false positive rate popping up. And this key finding essentially means is that we had better, if not equivalent, analytical sensitivity was observed again across all 7 sites when compared to what the claim is for this particular assay by the competitor, which is supposed to be 0.1%. Again, I do want to stipulate that this is not a head-to-head comparison study. It's just an observation and obviously in line with what has been projected by the competitor in terms of the analytical sensitivity that they observe with this particular assay. So another aspect that I'd like to highlight, essentially, and this is more from a qualitative standpoint, is that, again, from the experiments 1 and 2, as I've already mentioned, I just want to kind of reiterate, this is an off-the-shelf Bio-Rad assay. And you can see that there was no optimization that was done for the Digital LightCycler system. And what I think you can appreciate from these 2D plots essentially here is where we have the Roche Digital LightCycler on the left-hand side and the Bio-Rad here on the QX200 on the right-hand side. And I think what you can appreciate is that compared with the Bio-Rad plot, the Digital LightCycler has very much better cluster tightness and signal-to-noise ratio compared to what you can see here with the Bio-Rad system. Now, we do have an observation of slightly higher cross-reactivity, which is likely due to the fact that the system and master mix are different, right? As I mentioned, this was not optimized on the Digital LightCycler system, and we do think that there's an opportunity to do further optimization to actually reduce this cross-reactivity that we are seeing observed here on the Digital LightCycler system. So again, the key finding from this aspect of it, again, from a qualitative standpoint, is just to reiterate that there's an opportunity to port over assays that have not been optimized, have never been seen on the Roche Digital LightCycler, and there's a potential capability that you would not have to do a lot of optimization once ported over to the Roche Digital LightCycler, as demonstrated by this particular assay. And now I'm going to go ahead and move on to the final experiment. And in this experiment, again, we're kind of shifting over, and this is going to be a copy number variation as opposed to a rare mutant detection assay. And you can imagine that for a copy number variation assay, there's obviously going to be a really, really important for measuring very small differences, right? So precision is going to be highly important here. And so again, this is not using a Bio-Rad assay. This is using a NIST, the National Institutes of Standards, essentially published oligomer design for HER2 and the balancer, which is going to be EIF5. Again, EIF5 here in the FAM channel and HER2 here in the HEX channel. And what we're looking at here is specifically in terms of the analysis is looking for the accuracy and precision in being able to measure, the copy numbers relative to one another. And so one thing that I'd like to point out before we kind of jump into this is that the Digital LightCycler system is actually designed to really enhance copy number variation analysis, and for 2 reasons. One is that it supports 100,000 partitions per lane on the high resolution consumable. And the other aspect is that we have 6 channels that are available to allow for detection capability. You can imagine for copy number variation, it's really important to have multiple references, right, to allow for even more precise measurements with non-overlapping confidence intervals. And in that case, because the Digital LightCycler not only has a consumable that allows for more partitions to be kind of used for measurement purposes, it also has the capability of using more channels for more targets for reference purposes as well. And I guess I would like to kind of take this a little bit further and kind of indicate why this is really important. And I'm going to kind of showcase this with a little bit of a modeling here. So first I'd like to orient you, and here we have looking at on the y-axis here is the copies per genome. And in particular, we're going to be interested in discriminating 10 copies per 11 copies. This is a 10% difference, essentially. On the x-axis, we have the estimated input copies per partitions for reference. This is kind of represented by lambda. We're looking at anywhere from 0.02 to about 1.6, which is essentially the ideal in digital PCR. And then what's going to be depicted first in red is going to be looking at the uniplate. So the 10 copies is going to be represented by a closed circle, and the 11 copies is going to be represented by a triangle. And we're going to kind of showcase this in looking at both the uniplate, which has a little bit over 20,000 partitions, and the high-res plate, which has about 100,000 partitions, to really hit home why having more partitions is really impactful for copy number variation measurements. So to first kind of locate the uni plate, again, here we have 10 copies with the closed circle, the 11 copies here with the triangles, and what's emanating from both the circles and the triangles are the 95% confidence intervals. And ideally, in order to be very confident in discriminating between 10 and 11 copies, you want to have non-overlapping confidence intervals. And in this case, we can actually see that range in which of inputs in which you actually have non-overlapping confidence intervals is very small. Okay, so it's not a huge dynamic range, unfortunately. Now, this is in contrast when we actually look at the Digital LightCycler high resolution plate, which has, as I mentioned, 100,000 partitions. You can see that this area in which we have non-overlapping 95% confidence intervals between 10 copies versus 11 copies discrimination is over a larger range of inputs here. And this is kind of nicely showcased here by looking at this blue-filled center kind of region within this plot. So again, the take-home from this essentially is that the Digital LightCycler high-res plate can really provide that high resolution, that's needed to look at copy number variation across a multitude of DNA inputs. And so now here's that result from experiment 3, after kind of walking you through all of that. Here, what we're looking at is that HER2 ratio. Again, on the y-axis is the amplification ratio, the average amplification ratio. On the x-axis is your NIST sample. Now, these are 5 different samples at 2 different input amounts. The high input is 100 nanograms per reaction, and the mid input is at 20 nanograms per reaction. And I think what you could appreciate here is that across all 7 sites, again, color-coded here in this legend for you to kind of see, is that the measurements were accurate and consistent across all sites, and also the ratios were within the confidence intervals, as you can see, kind of across all 5 sample types in both inputs. Now, I do kind of want to point out that for sample number 1 and number 5, there are kind of residing a little bit on the confidence interval boundaries. And we do suspect that that's because these ratios are kind of likely due to the fact that in our assessment for this evaluation, we only use the one reference, right, the EIF5, whereas in the NIST references, they use 3 references for calculation purposes. So even though we're using only 1 here, we think that this accounts for why we do kind of see this a little bit more bordering on the higher end of the confidence interval, but still within the confidence interval, as well as for sample 5, kind of on the lower end of the confidence interval. But again, still within confidence intervals. And again, more importantly, the same measurement ratio across all 7 sites. So the other attribute of this, and again, the observed precision that I just kind of showcased to you with that data slide, is, as I mentioned, is really attributed to the fact that we have the versatility of using the 100,000 high-res consumable, again, which affords a lot of precision in terms of measurement. And this particular data slide is kind of speaking to the fact that with other kind of platforms that are on market, you don't have that advantage. And that's clearly seen here in this plot, where you have the 5 NIST samples kind of listed out here. on the x-axis, and on the y-axis, we have the relative uncertainty. So this is essentially measuring how confident you are in making a resolution difference between the copy numbers for these particular samples. And again, this is color-coded for you here in the legend with Roche kind of in blue, Bio-Rad, the QX200 in this magenta, and the QIAGEN, QIAcuicity in orange right here in the very bottom. And again, we're only looking at the more challenging input, which is the mid input, which is 20 nanogram input here. And again, what we'd like to highlight from this is that there is a gain in resolution when you're using the Roche Digital LightCycler system, because we have a consumable that's particularly tailored to this kind of application with this copy number variation. The other kind of aspect, in terms of not only being able to provide precise measurements compared with other platforms on market, is also, it was really straightforward to multiplex, actually, the HER2 and EIF5 on the Digital LightCycler system compared to other platforms. And just to kind of illustrate this, I just want to kind of show a qualitative data slide showcasing when there is an opportunity to duplex these two into a single reaction. So on the left-hand side, what we're seeing here is the Roche Digital LightCycler with EIF5 here on the y-axis, and on the x-axis you have your HER2. And again, your legend here for your clusters, your double negatives, your double positives, your single positives, and your single positives in this direction. And compare that with the Bio-Rad QX200, where you kind of have the capability of duplexing, but it looks like there is an inter-assay competition that is occurring here on the Bio-Rad platform when you try to duplex these two assays together into a single reaction. And it's likely due to the fact that there is, this particular reagent was not necessarily optimized for multiplexing, where in the case with the Roche Digital LightCycler, there is kind of optimization for the multiplexing to occur. Now you can imagine that this might be a little bit complicated in terms of being able to draw thresholds, which is what you want to do at the end of the day, right? At the end of the day, digital PCR very much is predicated on being able to classify positives from negatives. And this makes it a little bit challenging when you have a non-orthogonal double positive cluster that maybe kind of impairs ultimately quantification overall. And I think, just to kind of better illustrate this, even in 1D rendition, because you can also say, well, we can always, kind of threshold in 1D, you're looking at the same duplexed assay. Here on the left-hand side is the Roche Digital LightCycler system. Here on the right-hand side is the Bio-Rad QX200. You can see that overall, in comparison, that the Roche Digital LightCycler system actually has very distinct advantage in terms of the tightness of the positive cluster and the negative cluster, as well as the separation between the positives and the negatives relative to the Bio-Rad QX200. So making, the ability to actually threshold very unambiguous and therefore really allowing for precise measurements overall. Oh, I do wanted to note, just to make sure that it's a fair comparison, that in the actual evaluation design, because we saw this kind of inter-comparison kind of issue with duplexing essentially on the other platforms, that for the evaluation studies, the Roche Digital LightCycler HER2 EIF5 kind of duplex was done as a duplex in a single reaction, whereas on other platforms that were kind of looked at, they were done as single plex reactions, because we didn't want to kind of, we knew that there was going to be an issue essentially with duplexing them together. So just wanted to kind of point that out. So overall, from the evaluation side, I just kind of want to sum up, kind of the key findings of the evaluation study. And so essentially from experiment 1, what we kind of have learned from this is effectively that the Roche Digital LightCycler system really demonstrated consistency and reproducibility across all testing sites, right? This is internationally across, 7 different external sites, across multiple runs, both internally and, across all sites compared together. So very, very highly reproducible results can be obtained from the Roche Digital LightCycler system. And from experiment 2, we really were able to show that there is going to be at least an equivalent or better analytical sensitivity on the Roche Digital LightCycler system compared to other competitors across all the sites that were tested. And lastly, from the copy number variation assay, we have a distinct advantage, as I kind of depicted for you qualitatively from the 1D and the 2-dimensional plots, that we have really nice cluster tightness, better separation, and really nice resolution, especially for copy number variation where precise measurements are really needed. So kind of as demonstrated by these key findings, you can imagine that the high sensitivity and precision capabilities of the Roche Digital LightCycler system make it well suited not only for oncology applications, but a variety of applications, right? And some of which were just kind of listed here. We have microsatellite instability detection. There's also looking at pathogen load. NIPT is another really nice application that can be used that really requires precise measurements as well. And so for those reasons, we really strongly urge you to kind of make that leap to the new Digital LightCycler system from Roche and really unleash the true powerful potential of digital PCR. We do have a booth here. It's booth number 232, so please do come stop by. We do have the Digital LightCycler system there, and you can demo it if you would like to kind of look at it in person. And with that, I'm happy to take any questions if there are any. And thank you again for taking the time to come visit at our Spotlight talk today.